So in this section, we're going to look at cellular respiration, so how cells harvest chemical energy. So the big thing, and we'll review this one as we go along, is that we've talked about photosynthesis. So photosynthesis uses sunlight energy, okay, to take CO2 and water. We kind of use some sunlight to produce organic molecules, usually glucose and oxygen. Now, we need to, as consumers and even as plants, they'll need to take those organic molecules and harvest kind of cellular energy out of them. And that's ATP. So we need to generate a whole bunch of ATP. We'll also generate some heat. Okay, but the products that come out, think about it, we eat, we breathe, and then those will go into our cells, into our mitochondria, and out we come. We'll breathe out our CO2, and a little bit of water is there. So just by eating and breathing, you're generating a little bit of water to keep you hydrated. Not always enough, though. So, actually, I haven't given you an essential question. Let me go back here. I intended to. I'd rather just give it to you here so we have it. So, our essential question for today is, what's the process of cellular respiration? Draw on our owl here. What is the process of cellular respiration, and how does it power an organism? Okay. So that would be our essential question today. So let's take a look at some of these. Respiration, as it relates to breathing, and cellular respiration are not exactly the same. Okay. So we've got respiration versus cellular respiration. Respiration in the breathing sense refers to exchange of gases. That's all it is gas exchange. Usually an organism brings in oxygen from the environment and releases waste as CO2. So cellular respiration is the aerobic or oxygen requiring harvesting of energy from food molecules by cells. Okay, so cellular respiration is actually us getting energy, whereas respiration that we talk about is just for a whole like anatomy sense and physiology is just gas exchange. So respiration is required for cellular respiration, but they're not the same. Okay, so breathing, this is our respiration. You take in oxygen, you exchange and get out the CO2, but actually in the muscles is taking in that oxygen to be able to produce our little energy of ATP, okay, and to kind of produce the waste of CO2, which through respiration goes out of the lungs. So they're related, but not the same. Cellular respiration is exergonic, so you could think like exit, and gonic is something different, but it's kind of energy releasing. So the energy is exiting, exergonic. And it transfers energy from bonds of glucose to form ATP. Okay, so it's our energy releasing thing. Cellular respiration, here's a really good overview that's worth knowing, that cellular respiration can produce roughly 32, 34, 36, it depends on the book. Okay. It produces about that much ATP for each glucose molecule. It uses about 34% of the energy that's originally stored in the glucose, and 66% of it is lost as heat. That is what I think is amazing, is you can eat a glucose molecule, you're going to lose a bunch of heat. We can only manage to harvest about a third of the potential energy that's in a glucose molecule. Imagine what we could do if we could get all of that energy. Okay. So this energy conversion efficiency is better than most energy conversion systems. Only about 25% of the energy in gasoline produces the kinetic energy of movement. So technically our system's better than a car, but you know, it is what it is. Okay, so this is our equation. So this is our chemical equation. And so make sure you know that one. It'll be a little difficult to compare these two, but basically we're going to start with glucose and oxygen. Remember, this kind of comes from eating and breathing. and will release carbon dioxide, will form water, and will generate a bunch of ATP and eventually heat as well. The main parts of this are kind of these plus the ATP. All right, so ATP is also kind of can be a part of that equation as well. I got kind of interrupted there and had to pause the video and forgot where it was. All right, so your body requires a continuous supply of energy just to stay alive. You have to require energy. Right? If you're not sure if you require energy, watch one of those episodes of Naked and Afraid where they haven't eaten in like a week. Yeah, you'll see it. Okay. But you have to have the energy to keep your heart pumping and you breathing. These things require energy. Right? You don't think about them, but they actually do require that you have a baseline level of energy. So where we kind of get this connection to kind of health and diet is that the average human needs about 2,200 calories or kcals every day for energy. So about 75% of these calories is used to maintain a healthy body, and the 25% used to power physical activities. So a balance of energy intake and expenditure is required to maintain a healthy weight. All right, so you got to balance it, intake and output. So a kilocalorie, this is actually the same as a food calorie with like a capital C. So a kilocalorie is the quantity of heat required to raise the temperature of 1 kilogram of water by 1 degree Celsius. So we've had some form of chemistry, but we're on that one. Okay. It's used to measure the nutritional values indicated on food labels about kind of potential energy that's there. All right. So if you consumed, if you're about a 150-pound person, if you consumed food, this is how much you could burn per hour. Okay, so running, if you ran fast, I'm not that fast, but for an hour you could burn 1,000 calories. Okay, sitting and writing in an hour burns 28 calories. If you dance, about 5-10. So all of these kind of give you a range of in an hour how many calories you could burn. Okay. And this doesn't include the calories needed for body maintenance. These are just the activities. Okay. So how do your cells extract energy from glucose? That's going to be the process of cellular respiration that we talk about. That's kind of our main question. How do we extract the energy? It involves a transfer of electrons during chemical reactions. So that's how we're going to do this. Okay. Energy can be released from glucose by simply burning it. We could just light it on fire. That's lots of fun. That's something we like to do as scientists, but it's maybe not as beneficial. This electron fall happens very rapidly, and so the energy is dissipated as heat and light. It's not available to organisms once it's heat and light. And what a bummer. So cellular respiration is more controlled, and it's a descent of electrons, kind of like rolling down an energy hill. It's a very controlled and concise thing. Energy is released in small amounts and can be stored in the chemical bonds of ATP. All right. So I'm going to give you the brief intro on this one. I don't ask a lot of questions on this one, so if we just kind of get through and you don't write a whole lot of notes, that's cool. Let's just listen. But the movement of electrons from one molecule to another is called an oxidation-reduction reaction. They're also, for short, called redox. I told you, scientists are lazy. So I'll give you the shorthand on this in case you ever need it later, but we'll hardly use it in class. But if you lose electrons in a substance, it's called oxidation. So we say oxidation is loss, and then addition is called reduction, which seems counterintuitive. So we do say oil rig. Reduction is gain. Oxidation is a loss of electrons. Reduction is a gain of electrons. Oil rig. I won't ask you about that, but if you are really into science and see that as your future, you'll probably have to get to know redox and oil rigs. Okay. A molecule is oxidized when it loses one or more electrons. A molecule is reduced when it gains one or more electrons. That's all. I'm not going to explain to you exactly how all that stuff works. Just when we lose, so you're losing hydrogen atoms You're losing atoms and their subsequent electrons with them, so that's a loss of electrons. That becomes oxidized. When we gain atoms, we're gaining their subsequent electrons, so it's a gain, but it doesn't, it's a weird thing. Take OChem, you'll learn all about that. An important player, again, I'm not worried about the oxidation reduction. If you're like, I don't get this, cool, let's move on. Okay, so an important player in the process of oxidizing glucose is a coenzyme called NAD, which accepts electrons. Oh my God, it's another type of electron chauffeur. Okay, so this is like another electron, what do we say? I don't know. But do we all know what Uber is? It's an electron Uber, or it's like an electron chaperone, or any of those. Okay, it picks up electrons and takes them where they need to go. Notice, because we're going to have this guy, we have NADH here for cellular respiration. Quick thing, little side note. Remember, for photosynthesis, we had NADPH. So that was for photosynthesis. Okay, so I actually don't. I came up with this the other day. I'm very pumped about this. But to know the difference about who's happening in which area, in photosynthesis, we have NADPH. Notice it has a P in it. Okay. We have NADH in cellular respiration, and no P in that, no P in those. But for all intents and purposes here, they're going to function the same way as being a little chauffeur for our electrons. Never leave those little electrons unattended, not ever. All right. Basic thing here, you see NAD+. We'll pick up some hydrogens, but the big thing is the electrons. They'll sit right here, and they'll be chauffeured around. That's all we care about. Okay, we're going to look into the stages of cellular respiration. Since we're at 11 minutes, I would encourage you to maybe pause the video, go back, chunk your notes, write some questions out to the side, okay, take a look at it, highlight some stuff, and then come on back. So in our stages, we've got 3 stages. And this is one of those things that we'll do in class together. We'll go over it and make a big old table. But cellular respiration consists of a sequence of steps that are divided into 3 stages. Stage 1 is called glycolysis. Stage 2 is called really the citric acid cycle. That's what I'm going to call stage 2. I'm more concerned with that one. This one, this pyruvate oxidation, I kind of call it step 1.5. It's up to you how you do that, but I'm good with citric acid cycle. And then stage 3 is called oxidative phosphorylation. You're perfectly fine to call that the electron transport chain, because that's the major function that it does, which is a lot like the electron transport chain that we saw in photosynthesis. Okay. So stage 1, again, this is all the kind of terminology, and I'll show you some pictures as we go along, but glycolysis, it occurs in the cytosol or the cytoplasm. It begins cellular respiration, and it takes a glucose and splits it into pyruvate. So a little bit about this word, though, is glyco is kind of an old term for glucose. So this is going to refer to glucose. If you think back to lysosomes and things like that, there's the word lysis in it. So lysis. So we're talking about a process that splits. So this word is actually trying to tell you what it means. It says, I'm going to split a glucose. And that's it. We're taking a 6-carbon glucose. Those little circles are glucose. And then we are like fruit ninjaing it right in the middle. So, are we past fruit ninja? I don't know. Whatever. We are splitting it, so it's a glucose split into 2 3-carbon pyruvates. There you go. And that's all that one is. The second step is pyruvate oxidation. This is what I like to call step 1.5. We're just going to kind of dress pyruvate up a little bit. And we'll talk about that in a minute. Pyruvate kind of can't enter the citric acid cycle in the way that it currently looks. So we're going to talk about that. But both of these things take place in the mitochondria. And we're going to supply the third stage a little later with more electrons. The cell makes a small amount of ATP during glycolysis and the citric acid cycle, usually about 2 each. Okay, so that's a little bit what's going on there. Oxidative phosphorylation, also kind of known as the electron transport chain, is really going to take our electrons, and we're going to be able to basically do what we did before and get a whole bunch of ATP out of it. So we're going to talk about it. Those are the words. Let's look at some pictures. Okay, it's, again, the hydrogen ions. So this is going to be similar to the electron transport chain that we talked about in photosynthesis. Let's look at pictures. I cut myself off and hit the pause button. All right, so the picture, I think, is the best way to look at these things, and there's going to be better pictures too, but glycolysis is we're splitting a glucose into pyruvate. We'll make a tiny bit of ATP. We'll also have some NADH, and there's a different type of electron carrier, different type. Maybe NADPH is like a bus, the NADH is like Uber, FADH2 is like, I don't know, a taxi. Whatever, it's still going to make sure that our electrons get to their ultimate end product. We'll have pyruvate oxidation, or step 1.5 in here that we'll talk about a little bit later. We'll have citric acid cycle, it produces ATP. And at the very end, we get most of our ATP from right here. All right. Okay. So in glycolysis, a single glucose is enzymatically cut in a series of steps. 2 pyruvates are produced, 2 NAD plus are made to NADH, and there's a net gain of 2 ATP. So the things we're making are pyruvate, NADH, and ATP. Those are our products. Okay, so this is sort of what it looks like, is that I have a glucose and I want to split it. So I'm going to need to use an ADP plus some phosphates, and I can actually generate ATP from this because we get a little energy from breaking that bond. And there's also going to be some electrons that are let go of, and we can't let those electrons go by themselves. We already know that. We can't do that. So we have to add those electrons to an electron carrier so they can go off a little bit later. And in the end, we get our 2 pyruvate. That's it. Okay, that's the gist of what goes on there. Pyruvate formed in glycolysis is transferred from the cytosol into the mitochondria, where it can enter the citric acid cycle. But the thing is like it can't enter pyruvate. I'm giving you the gist of all this stuff. You can read it if you'd like to, but this is kind of what happens. So pyruvate can't enter the citric acid cycle. It's kind of like when you wake up in the morning and you're in your pajamas. Like, you could go to Walmart in your pajamas. That's not a problem. Nobody cares. It's Walmart, right? But you kind of can't come to school in your pajamas, right? It's not school appropriate. You get in trouble. You get written up. It's like, what? Okay, so we can't do that. So we've actually got to get dressed up a little bit before we go to school or go to work or whatever. So dressing up this pyruvate molecule is acetyl-CoA. Okay, so this is my little step 1.5. Okay, and basically what's happening is we're going to take the pyruvate, we're going to take some of the electrons, we get rid of a CO2, we get rid of the pajamas, we add on a coenzyme A, a tie, or whatever. We're doing a presentation today, so we're getting a tie and a suit on, we're putting on our, maybe it's Friday, we put on our service shirt, whatever it is, we're changing a little bit so we can be acetyl-CoA, acetyl coenzyme A, acetyl-CoA for short, so we can actually go to school in the citric acid cycle. Okay, that's the short. Okay, the citric acid cycle, it's also called the Krebs. So if you see the Krebs cycle, it's after this German-British dude, Hans Krebs. And he worked out this pathway because he, similarly to me, apparently doesn't have a life. So, no, I'm just kidding. For the love of science. Wouldn't it be cool to get a cycle named after yourself, though? We won't go into too many details here about the citric acid cycle, but kind of like what we saw with the Calvin cycle in photosynthesis, we have something coming in. This one's acetyl-CoA. It's dressed up. It's appropriate for work. It's going to lose a carbon dioxide. So this is one of the stages where we lose carbon dioxide. We also lost a little carbon dioxide in stage 1.5. Okay, so that's where our CO2 that we're going to breathe out comes from. We generate a few more NADHs, a few more electrons get picked up by an Uber, okay, to go do work somewhere else. And then at the end, we're going to generate a little bit of ATP. Those are the big things that are going to happen there. Okay. So during the citric acid cycle, the 2-carbon group of acetyl-CoA is joined to a 4-carbon compound. We form citrate. Citrate is degraded back to a 4-carbon compound. Really, the 2 CO2 are released, 1 ADP, 3 NADHs, and 1 FADH2 are produced. But remember that the citric acid cycle processes 2 molecules of acetyl-CoA. So remember our initial glucose is a 6-carbon sugar, and then we fruit ninja'd it in half. We did glycolysis. So we now have what will be 1 acetyl-CoA and then 2 acetyl-CoA. So this process has to occur 2 times. We go through the cycle twice. There's 2 turns. Okay. So our actual yield for 1 glucose molecule for the citric acid cycle is 2 ATP, 6 NADHs, and 2 FADH2s. Okay. I know, this is super complicated. So real quick, we'll do this before we move on to that last part. But after glycolysis and the citric acid cycle, the cell has gained 4 ATP, 10 NADHs, and 2 FADH2s. So to harvest the energy from our NADH and our FADH2 that have picked up little crazy electron children, these molecules have to shuttle their high energy electrons to the electron transport chain, very similarly to photosynthesis. Okay, so we are at 20 minutes. Take a break, pause, take a break, review your notes, then come back. Oh. I put this on here just to show you this is what we could go over. This is all the processes of the citric acid cycle we could do, but we won't. You're welcome. Know the other one. The final stage of cellular respiration is oxidative phosphorylation and involves the electron transport chain and this other thing called chemiosmosis. It does require a supply of oxygen. So the arrangement of electron carriers is built into a membrane that makes it possible to create an hydrogen ion gradient or a proton gradient across the membrane. Use the energy of the gradient to drive ATP synthesis. So hopefully this will be a review because it's going to be similar. Electrons from NADH and FADH2 travel down the electron transport chain to O2, the final electron acceptor. Somebody's got to pick up these crazy electron children, right, at the end of the school day. So oxygen picks up the H+, it'll form water. Energy released by these redox reactions kind of helps pump hydrogen into the mitochondrial matrix and intermembrane space. We'll look at it. Pictures are better. ATP synthase again. So it's the enzyme that makes ATP. Okay, so here's our big old picture, which is a mess. But basically, look, we've got NADH and FADH2. Okay, they don't really have any place to go. These guys are already energetic. They're already excited. So we put them to work. As the electrons go through, again, what's happening is we're pumping hydrogens against their concentration gradient. That requires energy. So we take the energy of the excited little electrons, the ADHD elementary school kids. We use their energy to pump hydrogens up their concentration gradient. So we're moving from the mitochondrial matrix into the intermembrane space. In a mitochondria, real quick, this is sort of what it looks like. Okay, so this part there is matrix. This is intermembrane space. Okay, because it's between, inter means between, it's between membranes. The matrix is this part, okay, all the way in the middle. So we're pumping it out here. That's where these hydrogens are going. Okay, so at the end of it, we're pumping all those hydrogens there. What we'll do is once those little electrons wear out, we're going to have them come back and pick them up. So those water, or the oxygen and the hydrogens will come together, pick up those electrons, make a stable water that can be used in the body. Okay, and just like last time, so we pumped all these hydrogens into the space, just like we would if we put all you guys in the LGI together. Eventually somebody's going to get bored or asked to go to the bathroom. We know that they're going to do that. So we let this little hydrogen, as it leaves that space, we say, can you do me a favor? Oh, wow, oh, thank you. Go ahead and take this ADP and this, can you just put those together? Oh, thank you very much. Okay, so as they leave, we have them do us a favor. Works the same way. Actually, doing it that route generates a crap ton of ATP. And that's how it's done. So we'll review those. I know that's a ton of information, a lot going on. We'll build some tables. We'll put it together. We'll try to make some sense out of it. So mitochondria. This is some thinking here. That's basic stuff. This is kind of our little tying it all together. So mitochondria and brown fat can burn fuel and produce heat without making ATP. Produce heat. So studies have shown that humans indicate that brown fat may be present in most people, and when activated by cold environments, the brown fat of lean individuals is more active. So this is a way that we can actually, without making any ATP, we can just generate heat, almost like, a nice way of burning the glucose so we can keep warm. Because there's no point in having energy if you're just going to stay cold all the time and have to shiver. Shivering will burn all that energy too. So that's just kind of skipping a step. Instead of burning all this ATP, let's go ahead and just make heat, and skip making the ATP. Keep the shivers down. Come on. All right, so energy payoff of cellular respiration. This is our basic review of what we've talked about. We've got a couple of steps. One is glycolysis. Our second one is more or less a citric acid cycle, but we've got to change the pyruvate just a little bit. We've got to change the close, make it acetyl-CoA, so it's our little half step. Then we have oxidative phosphorylation, which includes the electron transport chain. Total yields about 32 ATP. A number of reasons we can't actually figure that out is NADH produced passes the electrons across the membrane to either NAD plus or FAD, because FADH adds electrons further along the electron transport chain. It contributes less. Some of the energy of the hydrogen gradient may be used to work some of the other ATP in production. So there's just a lot of reasons. You don't have to write all those down. Okay, we're just saying it's not always perfect. I like this diagram. This is a really good one to kind of show you where it's going on. So glycolysis, splitting of the glucose, makes our pyruvate, happens in the cytosol, generates a couple ATP and NADH. We've got our making of acetyl-CoA, dressing that up, reforms some CO2 along with the citric acid cycle. And we've got NADH there too. Again, 2 ATP there. The end of the citric acid cycle, we form NADH, FADH2. All of these guys with electrons that they've carried are going to go drop them off at this playground, I guess, and let them really use that kind of energy to do work. It's probably like one of those new age ones where it's like we drop the kids off at the playground and they ride the little bikes and have a good time, and so they actually generate energy from that. Have you seen those? Maybe it's just me. I thought it was cool. Anywho, that's where they go, is they get dropped off here. They're going to ultimately get picked up by the oxygen and water their little families and go where they need to go and be all stable and happy. And us doing that with all these electrons generates about 28 ATP. So roughly we get about 32. Sometimes you'll see it as 34 and 36. It's okay. It doesn't really matter. We're just going to generate a whole bunch of ATP. All right, well, we can sort of do this one quickly. I know this is not ideal, but we're going to try to do this really quickly. Fermentation. Super, super quick. So fermentation is one way of harvesting chemical energy that does not require oxygen. Fermentation uses glycolysis only, produces 2 ATP, and will change NAD plus to NADH. Fermentation will provide an anaerobic path and will take NADH and recycle it back to the NAD plus that we needed to start with. Okay, so that's a little confusing, but we'll talk about it. So your muscle cells and certain bacteria can regenerate NADH plus through lactic acid fermentation, in which NADH is oxidized back to NAD plus and pyruvate is made to lactate. So what that looks like is, here's a glucose. We undergo glycolysis where we split that glucose into 2 pyruvate. Look, we've used the ADP and the phosphate to make an ATP. We had NAD plus, we make NADH, and we get our 2 pyruvate. Well, we realize, oh, we've started to make pyruvate, but we don't actually have oxygen available. Crap. So I've made this guy. I've made this guy, so I'm going to put him right here. I'm going to use the electrons here to actually change this pyruvate to lactate. So us doing that will get the NAD plus back. So this, and I don't exactly like the way that's drawn, what will happen is that NADH will go find another glucose molecule that it can separate and get 2 more ATP from. So it's just like 2 ATP at a time. It's a really like, oh boy, I forgot something important. Let me run to the gas station and pick it up real quick. It's a convenience thing. I don't have oxygen. I can't make that Costco run, but I can go to the gas station real quick and grab this or that. So some other things about this, lactate is carried by the blood to the liver, where it's converted back to pyruvate and oxidized in the mitochondria of liver cells. The dairy industry uses lactic acid fermentation by bacteria to make delicious cheese and yogurt. Okay. Other types of microbial fermentation turn soybeans into soy sauce and cabbage into sauerkraut. Okay. There's baking and winemaking. You've used alcohol fermentation, about the same process, but we're going to lose some CO2. We also make some, this is why you make bread, you'll put in like yeast, which will do the same type of fermentation, but it makes the bread rise with the little bubbles because it releases CO2. There's also a little ethanol, but particularly for bread, it's burned off with the heat. So same thing, glycolysis, we split the glucose, we have NADH and ATP formed. But when we get our ending pyruvate, we'll take this NADH that we've made, okay, we'll lose a CO2, we'll produce ethanol, and then this NADH can be used to go out to some other glucose and split it to get a couple more ATP, just to get a little energy going, but a little ethanol is produced in the process, a little ethyl alcohol. Okay. So there's some obligate anaerobes, which means they're obligated to work without O2. So O2, nope. Okay. They require anaerobic conditions. They're poisoned by oxygen. They live in stagnant ponds and deep soils. They're facultative anaerobes, which can make ATP by fermentation or oxidative phosphorylation. Yeast and bacteria can you really use depending on whether they have oxygen or not. They can be good. Like, oh, we have oxygen, we're good. We don't have oxygen, still good. We can do both. You actually, in your body, if you go running or sprinting too long, you don't get oxygen enough, your body will switch over to that lactic acid fermentation. So we too, kind of, not all the way, we are obligated to breathe, but, you know, we can sort of do some things without it. So glycolysis is the universal kind of energy harvesting process of life. It starts both fermentation and cellular respiration. The role of glycolysis in fermentation and respiration dates back long before oxygen was even present, when only prokaryotes inhabited the earth about 3.5 billion years ago. So that's some cool tidbits. The ancient history of glycolysis is supported by its occurrence in all the domains of light, its location within the cell using the pathway that do not involve the membrane-enclosed organelles of the eukaryotic cells. So it's kind of a side thing. There's one other thing I was going to tell you. Basically, carbohydrates, fats, and proteins, real quick here, and we'll talk about it, but basically we can use carbohydrates or fats or proteins at different areas in this pathway to produce ATP. So this is why we want to have a balanced diet. We want to eat a variety of foods because all of these can produce different levels of energy that we can use to make ATP, to power motion, to power your thought, because your thinking still takes energy. Power that heartbeat, that breathing, all that stuff that we need to happen. Okay, we're powered by a whole variety of foods. So that was really the only other thing I was going to give you on that one. Come on. Okay. Do we want to keep annotations? I do not. Anyways, that's basically where we end that one. So there's a lot to it. If you have questions, feel free to shoot me an email. Otherwise, when we get back to class next week, we're going to discuss it, make it work, talk about it. Remember, we have a quiz. But you got a test coming up the week after that. You get to this. I don't know. just put something on a piece of paper and give it to me. Well, you know, something related to this, something you learned, put it on a piece of paper, give it to me, get 5 points on your test. There you go. It's the easiest Easter egg I could think of. Put anything you learned from this on a piece of paper, give it to me. You got your 5 points on your test whenever we get to that in a little while. So if you have any questions, shoot me an email. We'll get to it.